Alrighty, for those tuning in at home and on shore, you can see we're just about arriving or getting close to arriving at bottom here. So the pilots are just taking an opportunity now to get settled in and tuned up before we start exploring this canyon. So stay tuned and we will start checking out some of what this canyon looks like very soon. All right, for those following along on shore, we're arriving on bottom here now. And you can already see there's quite a bit of biology in this area. So if you are tuning in from home, please feel free to follow us along in the chat. This will be a science-focused dive today, so we do welcome your participation. Video, this is Watch Lead. The scientists on shore are asking if we could get a zoom on some of these gorgonians here, or just any of... Thanks, video. Pilot, this is Watch Lead. The scientists on shore are asking if we could get a zoom on some of these corals here that we're coming around to. Great, thanks. Yeah, maybe we could start coming in on that, what looks like the white gorgonia on the, towards the bottom of the screen, has a couple of squat lobsters on it, or what looks like squat lobsters from back here. Yeah, sure. Thanks. Looks like we've got two squat lobsters here on what looks like a gorgonian, so a sea fan type of soft coral. Just lots of biology on this feature here. Our scientists on shore, Dr. Asako Matsumoto, is saying that this might be a precious coral that these squat lobsters are hanging out on. And it looks like we might have some Promenoid corals in here as well. As we zoom in nice and close on this squat lobster here, you can also see some of these coral polyps. Just really, really beautiful. So we're just coming in to zoom in on the base now of this coral. And our scientists on shore, Dr. Asaku Matsumoto, has identified some of these corals that we're seeing on this feature as Iridogoria, a black coral, some Chrysogorgia, some bamboo coral, Plexaurid, and crinoids are all here present on this rock formation. Pretty amazing array of biodiversity that we're seeing here just on this one feature in this canyon. It's pretty amazing. Looks like we've got a brittle star here on this coral, and this white coral that you're seeing at the top of the screen here has been identified as a Stoloniferous Octa coral, which is that white one, which is just in front of the brittle star. It looks like we got a cup coral attached to this rock formation right in the back there. And that yellow coral there to the right of your screen is a zoanthid. Looks like we've got a Promenoid in the upper middle between that precious coral that we were zoomed in on before and the Stoloniferous. Looks like we probably also have Haverocideris here. And this pink one, the upper pink one, is black coral. It's a nice little squat lobster there on the top of the screen, top left. Just taking a quick snap zoom on this squat lobster. And these yellow star-like corals on each stalk are crinoids. And we also had a feather star on that precious coral there, right next to the squat lobster that we just snap zoomed in on. So a really great start to this dive here as we come over the top of this feature. Lots of life down here. Already seeing some incredible coral biodiversity. So that spiral-looking feature there to the left of your screen has been identified as a Haverocideris. Just really incredible spiraling on the morphology of this coral. Looks like there's a brittle star wrapped around the stalk of this coral here. Some more squat lobsters in there. Oh, several brittle stars wrapped around the stalks here. And our scientists on shore are identifying this one as a bubblegum coral. There's also some feather stars on that bubblegum coral there. Just a lot of life on that one coral feature. I'll take a moment to introduce myself while we're coming in over the top of this feature. My name is Trish Albano. I am the Internship Program Coordinator with NOAA Ocean Exploration. Just filling in for Dr. Kim Galvez, our Expedition Coordinator, for this hour while she attends a meeting. So my role in the office focuses on managing all the different student opportunities that we have for undergraduate students, graduate students, and early career folks to get involved with ocean exploration. If you're a student who's interested in our office's mission and you think what you're seeing here is really exciting, you can visit our website at oceanexplorer.noaa.gov and check out our student opportunities page for internship and fellowship opportunities. So directly in front of us, we're looking at a precious coral with a purple soft coral at the base. And again, those squat lobsters are hanging out on it. We're going to go ahead and get a closer look. Oh yeah, now you could get a really close look at those purple soft corals at the base of this precious coral. This squat lobster looks like he's kind of burrowed himself in. Oh, nope, that's not a squat lobster now that we're coming into focus. It looks like there's one back there, but some sort of anemone. Yeah, Pilot, if we could maybe go ahead and have a closer look at that fan, that yellow fan in the background. Yeah, back left. And that left coral on the left side of our screen here is a bamboo coral. So we're just going to go ahead and come around to check out this yellow fan in the background. Yeah, it looks like there's a squat lobster hanging off of the left side of it. Thanks, Pilot. Definitely some sponges over here over the top of this feature. Looks like we've already got some brittle stars here and what looked like some crinoids over the top of this coral. Just going ahead and having a closer look at the base of this coral. There's a Promenoid coral here down at the base, and scientists on shore, Saku Matsumoto, has identified this as a Promenoid. Looks like there's potentially a little hermit crab right there with a white shell on the bottom of the screen, and another one of those squat lobsters there in the background. Directly in front of us, this looks like a Stoloniferous coral, and there's some white polyps on what looks like a actually dead skeleton. Yeah, you can see these two white polyps here. There we go. It looks like, look, is he he's got a shell. Yeah, it looks like we got a nice little hermit crab on these coral polyps here. So we've actually got 3 corals here in this structure. One is Stoloniferous, we've got a precious coral, and a Promenoid. So again, just a marvelous display of coral biodiversity on this feature. Clearly serving as a home to several different types of crustaceans. We're seeing hermit crabs and squat lobsters. Just a really great feature that we landed on today. Looks like we got a crinoid in the middle right of the screen. This is a feather star. And what looks like maybe a brittle star wrapped around its base. Looks like we've got a zoanthid here on the right, or what was the right just a second ago. Pilots are going to come around and try to have a closer look. Awesome, thanks. All right, so we're just going to zoom in on this coral here. And scientists on shore are identifying this colony in the back as a promenoid, but thinking there could be two different species here that we're looking at in front of us, the white and yellow, potentially two different zoanthid species. Yeah. It's a very round-looking squat lobster. There is definitely a hermit crab here on this coral in the front. Just some really beautiful crinoids there on the top of this coral feature. For all those tuning in at home, we have been on the bottom here for about an hour, touched down at a really beautiful coral garden-esque type of environment on this rocky outcrop here. Seen several different species. Oh, looked like something just came across the top left of the screen. Seen several different species of coral here, many squat lobsters and some hermit crabs as well. Pilots are just taking us around to the other side to have another look.
Ooh, that's a really beautiful looking kind of delicate coral, which leads me to believe it's probably a chrysogorgid, but I cannot say for certain. Just looking into the chat room, maybe someone can help me identify it. We have some beautiful brittle stars that are on this coral here. Even though it looks so fragile, you can see just how strong this coral actually is by being able to hold up these brittle stars. But granted, you know, in the water column, things have different buoyancy levels. So, ooh, Scott France says it's actually a Metallogorgia. So, great. Thanks, Scott. We have a beautiful view here. I'm sure Trish has already talked a lot about it, but we are seeing unstocked bamboo corals. We have a variety of different kinds of Octa corals, I think, that are right in view here. Quite a few dead stocked corals that have some like some small growths on it. We're seeing a cluster right at the crevice of this like boulder area here that there seems to be some coral stalk debris piling up in this area, but we still have a lot of beautiful live corals living here. In the back you can see that Iridogorgia, if I'm not mistaken it's Iridogorgia megaspiralis, but it probably need closer identification to truly identify it down to the species level. I'm also seeing various bamboo corals. We have beautiful yellow and white colors all around here, and purple I think also in the background. We see quite a bit of these crinoids as well. And as you can see, everything is reaching out far into the water column, hoping that these polyps are reaching out there, grabbing whatever is in the water column, this detrital material or this organic material that's being swept by the current. Oh, wow, that's a huge one back there. Yeah, it looks like that Iridogorgia megaspiralis also down there. So, yeah, I think pilots already heading over there. It just would be a really interesting seeing. Oh, it looks like we have a fish also just swimming right out of view now. It would be really great to get some close-ups of that large one.
Maybe Scott France or someone can tell us something about the bioluminescence of some of these corals. I know we've touched them in the past, and we've seen them bioluminesce.
And Scott's pointing out that several of these colonies look to be overgrown with zoanthids, so that's a really good point too, that not just because it has a color to it doesn't mean that that's the original living coral. It could have been something that adapted its way and utilized the coral skeleton as a way to also reach out into that water column. Oh, and it looks like there's something red also sweeping by just in front of that coral that we're heading towards. Maybe a shrimp of esque. Ooh, getting a good zoom in here on this Iridogorgia. Oh, you can see a little squat lobster hanging off there. Beautiful spirals coming off this, and you can see just faintly that the polyps are all looking quite happy. They're all standing outright. Really fine looking polyps. This is a beautiful shot. Thanks, Pilot. It's interesting to see that the top end of this coral has a lot of those branching effect that where the polyps are versus down, as you can see what's coming into frame now. This lower section of this skeletal structure is actually bare from original polyps of the coral. We can see there is even not just like schmutz and everything growing on it, but it's actually quite darker as well. It could have been that ferromanganese is starting to also encrust on this, which means that it has been quite a while here. We can even see another coral kind of growing in and around the base here. Pilot, could we potentially get some lasers? Thank you. Oh, we also have a fine coral just off to that left there. Yeah, exactly, coming into frame, center frame now. Really beautiful. I mean, if the two lasers are 10 centimeters apart, you can just imagine how large this Iridogorgia actually is, even though a good portion of it doesn't seem to be alive anymore, but still at that upper end of it is. So it could still be precipitating calcium carbonate and growing still continuously. So really amazing feature to see. Ooh, Les Watling says the height of each turn of the Iridogorgia magnispiralis is about 20 centimeters. So that we could also use that to determine how large this coral actually is. And I'm guessing at least 2, if not 3 meters. And Scott Friend said that at the base of when we were looking at the Iridogorgia, there was a white coral, and that's likely, by looking at the base, a corallium that's there. Hi, did someone just join the call?
Scott Francis called in for a little bit.
Hi, Scott. How are you?
I am doing very well. That was a really beautiful spot that you alerted us to and found. Thank you for that.
No problem. I hope Ocean Sciences is going well.
Yeah, it's a little bit dizzying trying to keep up with a virtual conference and all the different meetings and Zoom rooms and then this cruise as well. One of the things about our digital age, there's a lot that competes for our attention.
Absolutely. We're just going to get into reposition here before we approach the canyon wall again, or this ridge feature. But so far we've had such a great dive already. We came out right down on this large coral community. So we've seen quite a bit of different corals. I don't know, Scott, if you kind of want to reiterate what potentially we have seen already and what we're likely to continue seeing here.
Yeah, well, what I've heard, you're doing a good job. I see Asako Matsumoto is in the chat providing you with some help here. And I've only seen a few minutes, but what I saw, there were most definitely a couple of species of bamboo corals in there, the Iridogorgia magnispiralis that you've been looking at, that beautiful, beautiful colony, Chrysogorgia. We saw the Metallogorgia when I first chimed in there that had its characteristic single brittle star living in its center. I had noted, and you had noted, that there are zoanthids, which are essentially colonial anemones, these yellow polyps that overgrow, almost like parasites, a number of other invertebrates, including sponges and corals. And I saw some overgrowing some Paragorgia, but then I also saw a yellow colony that didn't get a close look at it, but I thought perhaps that was a stony coral, Analopsamia perhaps, so not zoanthid, but I didn't have a close look. What else did I see? There was something from the family Coralliidae, possibly a corallium. That's a group of what they call the precious corals. They have a very hard, smooth skeleton, but it is an octocoral that was growing at the base of the Iridogorgia. So some good diversity, just in that one little patch that I saw.
Awesome. Thank you, Scott. This is overall really nice feature to also look at. You can see within this area, we're on the southern edge of the West Florida shelf within the US EEZ, and you can see this just massive wall in front. If you look on stream 3, you'll see our multi-viewer screen, which has not only high pack and different options to see from the Sirius's view, but you will also see on the lower right-hand quadrant our blue view system, and that's a sonar installed system that helps the pilots navigate and work through the water column, as well as to help detect them of any geologic features or any other kind of features that might that the light might not penetrate first, but it helps give the pilots a lot of spatial awareness of what's going on before they can actually see it with the vehicle itself. So it's a great safety feature for the vehicle, but also great at detecting any kind of other features that we had. On Dive 2, if you were able to tune in, we had an underwater cultural heritage dive, which means that we were actually able to not only complete some engineering objectives, but also we came up on a shipwreck. So, and that was within collaboration of BOEM and Search, or the Bureau of Ocean and Energy Management, and Search, where they were able to provide us with coordinates, and we were actually able to find and collect imagery data as well as other environmental aspect data with it as well. And the Blue View system was really instrumental in being able to detect a lot of these shipwreck ideas and that shipwreck imagery, and the pilots were really able to fully observe and utilize the Blue View system to its maximum with the shipwreck dive. So that's just one of the great tools that we're able to have on board D2, and hopefully we'll be able to continue advancing this as it goes. You can see this beautiful geologic feature that we have here, this canyon wall here while we're scaling this ridge section here. You can see that a lot of it has been encrusted, so this dark features that you're seeing on the top, on the surface of these limestone features is relatively dark, and that means that there's been a lot of ferromanganese encrusting on this. So a lot of these minerals and different elements within the water column can precipitate, but usually over geologic time. So that means in the factors of either tens to hundreds of thousands of years, even millions of years. So a lot of these features that are here, as we know in geologic time, were for those factors, it might seem fast, but for the human aspect of things, it can take excessively long, ancient times, and it's so incredibly slow precipitating that we will not be able to physically see a lot of this happen within our own lifetimes, but we see this continuously happen through the geologic record. And it provides a great substrate for all these cold water corals to actually latch onto and colonize and begin to grow. So as you can see, a lot of these corals are reaching out into that water column that I talked about earlier, hoping to get a lot of that organic and detrital material that's mixed up within the water column. So not just the minerals and the elements are chemically within the water, we're also seeing marine snow, and these are these little particles that you see right now on the screen within the water column itself. That's a mixture of either decaying organic material or smaller phytoplankton material that the corals actually use to feed on. And corals, as well as sponges, and most sponges are filter feeders that are able to process this water through their filtering processes and able to consume it as well. So the advantage for these corals and sponges is to reach out far into the water column as far as they possibly can in order to be able to catch the most of this organic material that's within the water column there. And here we have another Iridogorgia. beautiful, just as the other ones. This spiral feature almost looks like fireworks. We're also seeing a lot of the white coral around it with some crinoids. I'm not sure if it's a corallium or something else. But you can even see in the background some dead stalks also around there. And also a black coral, the black whip coral that's on the left-hand side of the screen.
Yeah, Kim, from this distance, I think on the right there, that planar white colony with the yellow crinoids on it, I think that's a type of corallium perhaps. And then just behind it, there's a pink one that is maybe a different species in the same family. So maybe it's a hemicorallium or possibly a paragorgiid. I couldn't quite see it, but...
Yeah, thanks, Scott. We can try to go back there after to get a better viewing of this, but right now we're zoomed in on this black coral here, this Antipitherian, beautiful orange whip-like coral.
Yeah, so this is the one that probably the genus that recently got a new name. I think the name is Aphanosticopathes. We used to call it Sticopathes. And that new name is an indication that genetically it's not in the same family, taxonomic family, that we thought all of these whip-like black corals belong, but is in a different family, the Aphanopathidae. And so that's what results in that complex name.
Wow, I didn't know that it was potentially getting a new name, so thanks for that, Scott. And for those of you also tuning in, Scott and anyone else also in the chat room, we are open to taking samples for today's dive. So if you do see something that would be a great contributing factor to science, please let us know and we'll be able to set up and collect it. Pilot, can you hear me? There was a request to see the coral that is on the starboard side, right after this white planar one that's coming into view now. It looks slightly fragile right in the background view of this, coming into view right now. Slightly pinkish. Thank you. Yep. It's actually a really great description of that, a bonsai tree looking. Here we have quite a few brittle stars, and it seems all the polyps are contracted. And this, Scott was saying, is potentially a hemicorallium, right? Ah, yeah, there you are, Scott.
Yeah, sorry, I was studying it as we were getting closer. I think, is there a lot of barnacles on there as well, I guess, on the right side? A little hard for me to tell.
Yeah, I am seeing some barnacles.
Change.
And even gastropods, I think.
There we go. Put on glass.
I think they're moving to get a better view of it.
No.
Stay tuned.
Okay. I am not 100% certain if that's in the family Paragorgiidae or in the family Coralliidae. I'm kind of leaning towards Haverocideris, actually, but I'm not certain. And it looks like also the yellow one that's just to its left, that must be a Plexauridae, probably Paramaricia, which are quite common here in the Gulf of Mexico.
Absolutely. And Scott, you were saying Paragorgiidae or another one that now escapes me, but I just wanted to ask what the overall difference is that you think of that. Oh, squat lobster sailing down.
Yeah, the other one I said was Coralliidae. So those are two very closely related families, at least in terms of their genetics. People who tune in a lot will recognize bubblegum coral as a classic member of the Paragorgiidae. And the way their skeleton is constructed is a little bit different from many of the other octocorals that we see. And the coralliids have a similar kind of construction of their skeleton, but they are able to make it much more dense. And so basically, I don't know, it's hard to say when you're looking at an image, but it almost looks like it's, what's the word? It breaks much more easily. It'll shatter more easily than the Paragorgiid, which is more flexible, if you want. Brittle is not quite the word I'm looking for. There's another word. I can't come up with it. But the problem in separating them is that there are some coralliids that are pink, so we can't just use the color. But the small size of the polyps on this pink one, the way they're coming off the branch, does look to me more like a coralliid. Most of the Paragorgiids I'm familiar with, the polyp itself, which is the part that has the tentacles, can withdraw into the tissue, and so you sort of see a smooth overall appearance. And that's not what I'm seeing on this one.
Wow, thanks, Scott. Yeah, right now with all the... Oh, go ahead.
Yeah, sorry, Kim. I was just going to say, just like the engineers, the pilots, and the crew, having to reset themselves every new season, same for the biologists. I have to reset myself about what I'm seeing and remind myself and remember what are the characteristics that I need to look at to separate this from that. So it's still early in the season for me.
Absolutely, and that's why we're calling this a shakedown. I think it's helping all of us get back into the groove of the field season and hopefully move slightly more towards anything normal. Getting a great zoom in on here. And you can see the base of this coral is quite large, and the main two branches or stems of this coral are actually quite dense and thick, whereas the polyps in comparison look almost fragile and really delicate overall. We're seeing quite a bit of brittle stars in this area as well, and there's a small stalk crinoid.
Yeah, this is a great image. So, right now, the polyps have contracted, and so you can't see the tentacles on any of them. They just look like these little wads of gum, which is pretty characteristic of a Paragorgiid. So that's the way I'm going to lean now. There's two of them. Actually, the two things that have their tentacles extended look to me like anemones. Huh, that's interesting.
Yeah. Do you think those are anemones or potentially an open polyp?
Well, if it was a coral polyp, it would have only tentacles. They would be pink, and at least in my view, I can't see the pinnules on them. And that's why I was suggesting something else.
Oh, okay.
It does not have a very good view. So it would be interesting for us, if we're in the position, to keep going to the left and look at the white planar colony just next to it and see how the polyp structure compares to what we're seeing on this one.
Pilot, did you get that? Slightly to the left to that white coral that's right next to it, adjacent, and just to compare the polyps. Thank you.
So for everyone watching, what we want to see is, do the polyps seem to be arising in the same way from the branches as on that last colony? Certainly, they're all exposed on this one.
Yeah, I wonder what the difference is between the two corals right next to each other, how one is so closed and the other one decides to open all the polyps, but yeah, they all tend to generally either facing down or to the frontal part of this coral.
Yep. To me, that's kind of a classic that I see on the coralliids. They all face one plane. Yeah, that other colony was just partying too hard last night. It's got to sleep it off now.
Probably. Got its fill. Oh, these are beautiful polyps. They look so delicate.
You know, we're often saying in science, the more we learn, the more questions we have. And sometimes the more I look at these colonies, the less I think I'm able to separate them.
I totally hear that, Scott. The more we learn, it just brings up more questions. That just leads to more exploration. So
I'm going to stick with the family Coralliidae for this one. I see Les Watling is suggesting maybe this is Hemicorallium beari. And I'm going to say the other one, that pink one. Something about that.
I will double check.
It just also says Coralliidae to me.
Scott, do you think it would be worth getting a sample of either one of these corals, or are they frequently seen?
Yeah, so I think they're pretty well known. You know, any sample's a good sample, but the hesitation I'm having in identifying them isn't necessarily that they're not well known. It's just my ability to know this particular family, and just, you know, the layoff of several months and not being used to doing this.
Absolutely, no worries. Thanks, Scott.
I don't have a compelling reason to suggest it be collected, but
no worries. We'll continue transecting this area here. Hopefully we'll find some different or new species that could potentially be a collection. But we're getting a really wide angle view again of this geologic feature, and you can just see overall how these corals are really taking advantage of being able to reach high out into the water column here, and just the overall different species that we're seeing. There's also quite a bit of glass sponges hanging off on some sides of the rock here. And if you look on stream 2, you can get that view from Sirius and with D2 in it, and just see exactly the grand scale of this huge canyon wall that's here, of this ridge that we're transecting currently. And you can almost make out some of the geologic features that are here. So we're seeing what looks like these long parallel blocks, and that could actually be different times of formation of the limestone that helped form these canyon walls. We're seeing a lot more of this Iridogorgia here.
the wonder of modern technology and telepresence. I just got an email from a former PhD student of mine, Jana Toma, whose class from marine biology class is watching the dive and enjoying this. And we had seen that Haverocideris, and Jana's dissertation work focused on Haverocideris and Plexaurids. So she's well familiar with those.
Wow, thanks. Well, big shout out to them. Thanks for tuning in and watching. I mean, it's a really great way to be able to share this with the community, and just to go, like you said, as far as advances technology has gone, that this telepresence capability, you are all at home able to see this live with us, while portion of us, a fair few of us, are out on the ship and being able to do this, but we're so happy that we could share this all with you. And we really rely on scientists at home, their expertise is to chime in on the science chat. As you heard earlier, if you were able to tune in, Not all of us on the ship, we can't host every expert on here, so there is a limited capacity, but being able to touch with everyone online and be in communication really helps us expand not only the scientists' knowledge on board, but also the general community as well. So we greatly thank all of your contributions towards our commentary, as well as in the chat room, being able to help us identify things, whether it be from geologic standards, biologic, and also including the water column in this area. So looking forward to all that has to come, not just for this dive, but also putting in a plug for the rest of the field season. We have quite a few mapping cruises, and then we currently still have planned the Mid-Atlantic Ridge. So stay tuned for those. Well, they'll be within June and July of this summer.
I've got a question for Shoreside. On this expedition, we've been testing out a low-light camera during midwater, and I was wondering if we have an idea of how many of these corals have any bioluminescent properties.
Ooh, that's an excellent question, Video. So Scott did mention earlier that some of these corals do bioluminesce, especially when agitated. So I don't know, Scott, if you're on the line, if you'd like to elaborate on that.
Yeah, so I heard that question from Roland. It's a really good question, and there are research groups who are actively studying that, and it's actually quite remarkable the number of different benthic invertebrates that are capable of some form of bioluminescence. And just so everyone knows what we're talking about, bioluminescence is a process whereby using enzymes enzymatically, organisms are able to produce flashes of light. And it's usually a bluish sort of light. And that's different from some other things like iridescence and phosphorescence. And so it is a chemical reaction. It requires a substrate. It requires the use of energy. And so organisms usually are using it fairly judiciously. And Roland had mentioned the midwater there. So up in the water column, there are several organisms that, classically like some fish, that are able to use bioluminescence as a method of signaling or predator-prey interactions. It's a different question. Why would these organisms that are living attached to the substrate that don't have eyes be using the bioluminescence? We're not quite sure about that, if it's some sort of defensive technique or what, but absolutely we have seen, for example, in some of the bamboo corals where under certain conditions, if you touch them, if you physically touch them with the manipulator, or we've seen it on some of the specimens that have been collected and brought back up onto the ship, we can go into a dark cold room. You can see that the polyp will glow blue, and then that signal is propagated along the tissue so that the polyps on either side then start to glow blue, and the first one goes back to not glowing. And in that way, it looks like there are these banded flashes of light that are extending from one position where you have touched the coral running along the tissue. Now, why? That's the challenge. Scientists have to come up with some good experiments to test, what might be the physiological reason for that bioluminescence.
Wow, thanks, Scott. Oh, yeah, we're actually seeing quite a bit of coral debris. So this rubble, that's kind of the breakdown of the leftover skeletal structures of any of these corals. Now, if you tuned in yesterday, ooh, was that a sea urchin? Can you possibly zoom back in there? Just slightly further up, up and to the left. It was just this tiny little white ball. There, just coming into screen now. Chris Ma, I see you in the chat room. Would you like to call in?
While we're waiting for Chris, I'll just say I got an email back from Dr. Toma, who sent me a photograph of the class watching, and I neglected to say before that they're all at Mississippi College. So thanks for tuning in. I'm glad you're enjoying this dive and learning something, and you're lucky enough to have a teacher who's an expert in these deep-sea corals, so good for you.
Absolutely. Thanks for tuning in with us.
Scott, that's awesome. Could you forward it to us here on the ship?
Yeah, will do.
Thanks, Scott. That was a really interesting looking sea urchin. Did we happen to get lasers on it for potential size? Could we? Ooh, really small, actually.
It's so tiny.
Ooh, maybe 5 centimeters, 4 centimeters, including the spine structure. So if we're looking only at the disc in itself, it's probably only 2 to 3. So really small there. Thank you, Pilot.
Mike Vecchione just made this interesting observation in the chat room that if something is eating a bamboo coral that lights up, that's going to attract a larger predator, which is true. And certainly I have never seen bioluminescence when I've seen some predator like an urchin or a worm or something feeding on the bamboo coral, but the timing is different. They can't constantly be bioluminescing. There just wouldn't be enough energy for that. And we may not have the appropriate light settings. We may be missing some bioluminescence that's occurring when we have the bright lights on.
Yeah, absolutely.
Chris just called in.
Yeah, this is me. I'm trying to hunt the thing that we just saw. We actually saw one of these during Alan's tour as the science lead on the last expedition. We have it to a genus, and what's it called? Haverocideris. It's a fairly rare deep sea urchin. It's really fragile. I've seen the fragments of the species of the specimen we have in the museum, which I think the last time we collected it was via trawl from, I want to say, the 1960s or so. Those things, like a lot of deep sea urchins at this depth, are so fragile that picking them up could literally just shatter the whole thing. So the last time we were out, we collected one, and it will probably be a great value to people trying to understand, number one, what that species is, and number two, why the skeletal morphology is so unusual. But we really didn't know very much about it, other than the fact that it occurred in the Atlantic and that it had this really unusual morphology. So every time we see one of these, it gives us some insight into, how it lives, because when you collect a lot of these specimens historically, they're trawled. So we don't have any context as to, how it lived or why it appears the way it does in order to survive in its habitat. And so, granted, sometimes the adaptations are so bizarre that it takes a little bit of out-of-the-box thinking to get there. And like I said, it took us a while to figure out what this was, including conference with some fairly specialized scientists. So it's always nice to see them alive and to see what they're doing, especially if they're eating or whatever it is they do down here. And this, the habitat for these has been, I think, pretty consistently on these rocky surfaces. So that's good to know. Anyway, thanks for another occurrence data point for my urchin colleagues in the future.
Absolutely. Did I potentially hear that? Would this be worth collecting to try to, if it's like an entire collection, or do you think it's too fragile for a potential sample?
They got one last time. We just collected one. Okay. If you guys are hankering to use the suction sampler to sort of practice, I'm all for that. But But I think on the whole, you could go on. We have, it's always nice to have another intact specimen because, like I said, the specimens that we have for this species are all fragments. They're, the pictures and the specimens that I've seen are literally just pieces. But that said, it's probably something known. It for this area and for that genus, known species. So it doesn't adhere to the typical Okeanos guideline for collection. But, given that this is a practice dive, and who knows what kinds of weird things this one might reveal. It could be could be complete. I mean, this has happened, simply because this kind of resembles Haverocideris. It doesn't mean that it is. So it could potentially. I mean, I'm not an urchin expert, so for all I know, collecting this could result in the discovery of a new family. So if and it's quite small, if you have the time and the inclination, then I think it's certainly worthwhile. But but on the surface, I think we could forego it, especially since I can't say definitively that it's something. I mean, it's worthwhile, but I can say definitively that it's unknown.
Sounds good, Chris. Thank you. Pilot, would I guess the ball is also in your court there. For science, it's not truly a major undiscovered species potentially, but if you are in potential to sample and would like to, it would definitely benefit science. Perfect. So, Chris, what would be the ultimate preservation? We are. They're setting up for it and making sure that all of that the needs are being met by the ROV, just to make sure they're setting up appropriately to get the best angle for it.
Okay.
I did want to ask what the best way to preserve this is.
A relatively simple 95% solution would be fine. Alcohol.
Perfect. Thank you.
Thank you.
Could you possibly repeat the potential ID that this specimen would be?
Certainly. Habro, Habro, Cidaris, Cidaris.
Thank you.
Oh, you're welcome. Are those just some brittle stars off to the right there?
They look to be on the base of that little crevice there.
From here, I wasn't sure if they were sea stars or not.
Not sure. After the collection, we can get a snap zoom.
Yeah, no worries.
I'm pretty sure they're brittle stars. One of them may have been a Bersingid.
Ah, okay.
It does not want to let go. All right, maybe that urchin lives to fight another day.
Lesson number 1 in the deep sea. Just because it looks fragile doesn't mean that it is. That's fine.
Thank you so much for trying, Pilot.
Thank you, Pilot.
Yeah, I would say, there's something instructive here. We've been, doing this work for more than a decade and learning more about what are the tools that we need to sample. And that's one of the reasons that we have this suction sampler on the vehicle. We didn't used to have a suction sampler, and because there are some things you simply can't pick up with the claw, or the claw was modified so that it could cut instead of just break, for example. This urchin is sitting in a pretty tough little spot there on a rock, and so it's really hard to get to. And so maybe we need to develop other tools for making the sampling easier if we're trying to get things in these sorts of situations. But it's all about the science and the engineering and learning as we go. Another good use of the shakedown.
Absolutely. Thanks, Scott. We're currently, while we're settled down, we are testing the temperature probe just to make sure that's also functioning properly.
And before we go, can we just get a little view of the orange colony on the right side at about 3 o'clock? And I suggested that, unless you've seen it already on the diet, then that's fine. I suggested it might be Storopathes, but another alternative is that it's Swiftia. So just a quick look would tell us the difference.
I will reiterate. Thank you.
Thanks. I'll be watching. Next time a starfish pops up, I'll definitely chime in.
Thanks, Chris.
Thank you. Bye-bye.
And Kim, can I follow up a little bit on the bioluminescence while we're doing this, or do you want to describe more about what we're doing?
I'd like to go over really quick what the pilots are conducting now, because I also have a question for them. So just for everyone at home tuning in, we tried to collect that sea urchin that's almost dead center of the screen, but with unsuccessful efforts because that sea urchin just did not want to let go of that rock. Currently, we are testing just overall the processes of taking out the temperature probe, also testing that, the functionality, making sure it's retractable and can come out when needed. Again, shakedown cruises, like Scott mentioned, this is all part of that. So we want to make sure all of our objectives are complete, all the systems are groomed and ready to go for the overall field season. Pilot, before we do take off, there were one request, one from me and one from a scientist. If we could possibly take a Niskin sample here, because of all the beautiful coral would be around, it would be great for eDNA. I'm ready. Yes, so right at 3 o'clock, there is a small little bushy coral, if we could get a quick zoom on that. Thank you so much.
If I said 3, I meant... Oh, yeah, 3, yep. Sorry.
No problem, Scott.
Well, where does the clock work?
I don't know, time is irrelevant nowadays.
Right, it's all digital. Okay, yeah, 100% that is a black coral, Storpathes. Thanks, that's all we need.
Great, thank you. Getting a quick snap zoom, it looks so nice. And Scott, do you want to talk quick about what makes a black coral so distinct with its tentacles and polyps?
Sure, so most of what we're seeing down here are Octa corals. These are all cnidarians. They're all related to jellyfish and sea anemones, and they all have the classic nematocyst, which is a stinging structure that they use to capture their prey. So they're all soft-bodied gelatinous, but the corals have a hard skeleton, and so those soft-bodied gelatinous tissue, what we call polyps, sit on the skeleton that they construct themselves. And the Octa corals and the black corals lay down a different kind of skeleton. They're both basically mostly protein, maybe with some amount of calcium carbonate that's embedded in it, and that varies among taxonomic groups. So we were seeing earlier the corallium that has quite a lot of calcium carbonate. The bamboo corals have quite a lot, whereas the things like that yellow fan you see over there, the Paramyricia, they're almost entirely protein. So that's one thing that distinguishes them. Basically, the protein is gorgonin in the Octa corals, and it's antipathin in the black corals. It's much harder. And that black coral skeleton is typically black or very dark colored, and so the coloration would be different. And then if we were able to see underneath the tissue, you would see that the skeleton has spines associated with it. And the spines can be quite complex. There's a lot of diversity between species, and it's actually the main tool used to identify the taxonomic groups of black corals. And then the other thing is if you look at the polyps themselves, so the soft tissue that we can see, the octocorals, the easiest thing to tell them apart if you get a good view is that each polyp will have exactly 8 tentacles, and those tentacles are what we call pinnate, which means they have little side branches on them. So they almost look like little combs or feathers, if you will. Whereas the black coral polyps, the tentacles are smooth, and there would be 6 tentacles associated with them. So those are the sort of primary characteristics. And then there's some other variability that depend on what kind of black coral you're looking at or what kind of octocoral you're looking at.
Awesome. Thank you, Scott. So right at center, there is a bamboo coral just slightly south of dead center. Is likely bamboo coral. We can get a quick snap zoom just to confirm. An easy way to confirm that are those black nodes that are seen periodically throughout the branch of the coral. Oh, I'm actually not seeing any. I don't know if anyone is tuning in, if they can confirm whether or not that they see any. Or the pilots are really... Sorry, go ahead.
What were you looking for?
Potentially a bamboo coral that could be agitated for testing some bioluminescence.
Oh, I see. Yeah, I mean, that absolutely could be one. You would have to, turn out all the lights and then run the manipulator over it. But that would certainly be a good candidate.
Do any of these other corals?
Did you get that, pilot?
All
right.
Bioluminescence?
Do you want to go ahead and test it out?
Yeah, to directly answer Roland there, these could be ones of bioluminescence. I can't give you a 100% guarantee, but in no case could I give you a 100% guarantee a priori that this one will or this one won't. But this one is a good bet.
Sounds good. Thanks, Scott. Yeah, nothing is ever guaranteed, but we can try. All right.
So Tim, a little bit more about the bioluminescence, because I may have mischaracterized what Mike Vecchione was saying in the chat room. When he was saying that maybe that lighting up will attract a larger predator, I think what he meant was that if there was a predator on a coral that is causing the coral to bioluminesce, perhaps that's like an alarm that brings in a larger predator to eat whatever is disturbing the coral and not the coral itself. So that's the burglar alarm idea. And then Les Watling suggested that perhaps bioluminescence could be used to attract some small zooplankton that then the coral could feed on. So these are a couple of the ideas that would need to be tested.
Absolutely. And I mean, part of doing that is being able to observe a lot of these kinds of features in situ, in the water column itself, in the benthic community itself. So this imagery that we're able to get is actually vital to determining also how behaviors are seen, not just within the corals in itself, but also the organisms that interact with the coral, like you said, like if there is a predator coming to eat them, or crabs or squat lobster or fish or anything, or even jellies that interact with it. So it's a really great feature to see, and any predatory behaviors are really great lessons to learn. We were not able to set up properly for that specific bamboo coral to test it out, but we are on the hunt for another one. So feel free to let us know if you do see something that could be applicable. Right now, the pilots are getting ready for a change in watch, so hang tight, and we'll make sure that we get all set up once they're all settled in.
Okay. Kim, what I can say is where we have seen it is in some of these large bamboo coral whips, so I would say those are good targets. We're pretty fortunate today. There's another class that's joining the dive. This is Tara Luke's class from Stockton University in New Jersey. She has a deep sea biology class that she's running, and they're also following along, and they have some good questions. And there's one here. Are the body temperatures of these corals higher than the surrounding waters? And so that's pretty unlikely. They are, quotes, cold-blooded in terms of their metabolism. But I don't know that anyone has, actively studied that. Certainly, at some measurable level, I suppose, when the physiological reactions, metabolic reactions are going on, heat can be released, but I suspect it's really not enough to make a noticeable difference. So that means that the coral should be existing and surviving at whatever the surrounding water temperature is. And Les Watling had noted earlier that relative to this community where we typically see them, the water temperature here today is actually a little bit higher. Usually we see this at 2 or 3 degrees, and it's about 4.5 degrees, 4.3 degrees here, Celsius.
Yeah, thank you, Scott. That was a great synopsis. And who knows, maybe in the future there'll be a technology where we'll potentially be able to detect maybe those minor difference in temperature changes and including detecting metabolic changes within the corals in itself. So, I mean, as we know in standard body chemistry, it takes energy in order to consume things and be able to process it within the system, as well as precipitate things. So that means also our skeletons and also the same kind of skeletal structure that corals have. So, right now it's just so finite and so fine-tuned that maybe we don't have the equipment right now in order to observe a lot of these things. But who knows, in the future, some of these engineers, maybe some of those in that classroom from Dr. Tara Luke. So we're always looking for great scientists to come out and come up with the latest and greatest technology that can help us improve ocean exploration. So we look forward to potentially having one of you guys come up with that kind of technology.
Yeah, absolutely. There's definitely a lot of interest in the effects of temperature on marine organisms and deep sea organisms, especially as we, confront the reality that the Earth's warming and the oceans are warming, and what is going to be the impact on organisms. And in places like hydrothermal vents, which are areas where hot water is coming out of the seafloor, and there are organisms that are living around these seafloor vents. There's been a lot of work on using various kinds of thermistor probes to look at what is the ambient temperature that the organism is in relative to the surrounding water. How does that affect their metabolism? Temperature's got a very important effect on metabolic rates, on development rates, how fast an organism grows, how well it can reproduce, and so on. And so temperature is really critical. So I agree with you, Kim. We have some talented students, some talented engineers in the future can design the probes to be at the scale of individual polyps. Then we can actually answer that question. Measure the temperature of the probe in situ. That's the challenge, right? Down here on the sea floor, not necessarily in the lab, which would give you some information, but different information.
Thank you so much, Scott. All right, new pilots have come in, settled in, so they're giving their hellos, and we'll continue to look on. Potentially this bamboo coral right here could be a possibility if we want to try to agitate that. We have currently on D2 a bioluminescence agitator that we have used for the water column, and it resembles quite a large tennis racket that was utilized when we had some midwater objectives to complete. So that we were able to have that out in the water column in front of us and test it also with our low light camera. So, pilot, do you feel comfortable setting up for potentially testing this coral? So just any simple interaction with the coral could help promote any bioluminescence. So however you feel comfortable with it, either with the manipulator arm or by other means. I don't know, maybe if blowing out of the suction sampler could also be something, if maybe the manipulator arm might be too strong, but essentially set up with the low light camera and see what we potentially can find.
Yeah, Kim, my advice would be really all you want to do is, if you just took the back of the manipulator claw and then just kind of run it along, like you run it along the colony, like you would if you were running your finger along the teeth of a comb. That's what you're doing. You don't have to grab it or anything like that.
All right, great, thank you. Pilot, did you happen to catch that from Scott? All right, thank you. Thank you.
Yeah, I think we've done something like this before. It might have been the Hercules. Maybe it was a Deep Discoverer, but I think, we sat down, got the lights off. We sat down, put the claw in position, got the lights off, and then just moved the manipulator where we thought was the appropriate direction for it to intercept the coral.
With this new Canon ME20, we don't have to have the lights off completely because it does pick up the lights really well. So the pilot should still have a view of the coral as we do the test.
So, Roland, is there a separate camera to detect this, or is this something that we are going to see on shore? Or?
I will change stream 3 to be the Canon ME20 full screen.
Okay. Thank you.
Hey, Scott on shore. This is Chris piloting. I think I heard take the back of the manipulator claw and running it up and down, maybe just the side of this coral to see if that would work.
Yeah, exactly, Chris. So, I don't think we have to grasp it, and it may only require, just going once. You don't necessarily have to go up and down, and so I would say, go up, choose a direction, go up, go down, whatever's easiest for you, see if we see a reaction, and then if we don't, then run the manip in the other direction.
Okay, copy that. All right, we'll get set up for that now.
one more thing. It shouldn't matter if your claw is in the foreground or the background of the colony, whatever's easiest.
Okay, great. Thanks, Scott.
And for those of you tuning in, you want to look at stream 3 for our low light camera operations. I'm detecting some bioluminescence.
Yeah, I see it too. It's like on the tips of the polyps, maybe.
Wow.
There we go. So the polyps themselves are lighting up, but not the tissue between them. You can see that blue light, very characteristic of bioluminescence. That's really cool. And you see that it isn't persisting very long. And unlike what I was describing where you can see the flash propagate along the colony, I'm not seeing that. It's only where you're actually intercepting that colony and getting the physical connection. It's like a colony of fireflies. It's the same process.
Wow, look at that. What comes to mind is when you run your fingers down a piano and you're seeing the corresponding lights come out.
This sort of reaction, in my mind, isn't to attract prey to the coral, but certainly could be to attract predator to whatever is bothering the coral. So perhaps B2 should be careful here.
But
well, that's a great camera addition. That's really nice because we can clearly see in a manipulator at the same time that we're seeing luminescence, and we can pinpoint that luminescence to the polyps themselves.
absolutely, this has been a great view. That was really great. Ending the commencement of tickling the coral. Thank you so much, Pilot.
Well, yeah, I mean, science in action, right? We had a question, we had hypothesis that perhaps this type of coral was capable of bioluminescence, and we thought it would be due to physical contact, and then we tried the experiment, and there we go. And so the next set of experiments we would want to be doing is, maybe why, how long can it keep luminescing? It looked to me like we could go back and study the video, but were the same polyps repeatedly lighting up, or was it always different polyps? How much energy is there? Very cool. looking at this skeleton that's lying down just to the right of center. Looks like a candelabrum. This is the first one of these that I've seen today. You should tell me, Kim, and others. But that's a bamboo coral, one that Les and I are actively working on describing. That's got that very characteristic growth pattern of a single stalk that then gives rise to lots of these little side branches. So interesting to see that here. There's a dead one.
Thank you, Scott. And just to let everyone know, this is Anna Lenish. I'm along with Trish Albano here. We had a bit of a watch lead shift just as Kim stepped out for a minute. And so with that candelabrum, is that, even though we see it dead here, is that something that we would see in the area alive, or do you think it potentially drifted from a different location?
Yeah, it wouldn't have moved very far, and that's why I'm so interested in seeing it, and why I asked the question, has anyone seen any of those earlier today? I will need to call up my map. We've got a pretty good map of the distribution of this species, and I honestly don't recall if we have ever seen them in the Gulf of Mexico before, so I'm going to go check that right now, and I will get back to you. But,
wonderful. Thank you.
Anna, just to follow up. Yeah, just to follow up on that, that colony had to have come from very close around here, so I don't know when it died and toppled over, but it means it is definitely now in the Gulf of Mexico. It just would be nice to see a live one.
Right. Okay, well, that is great to know. And yeah, any information about the candelabrum for this area, I don't believe we saw any earlier today on this dive, so seeing one now is definitely the first time for this location here, at least on this dive. But yeah, knowing if they're more common present elsewhere in the Gulf of Mexico, or if this is something, would definitely be information that we would love to know. So thanks, Scott.
Right, so I just pulled up the map, and this would be the first observation from the Gulf of Mexico, so that would be very cool.
Wow, Scott, thanks so much. That's really exciting to hear.
Wow, another
Looks like,
first here on the Okeanos.
Sorry to talk over you there. Yeah, this is looks like a predatory starfish feeding on a Chrysogorgia.
All right, and we've got Chrisma identifying this predatory starfish here as Evaposoma.
And I don't know if it's the angle that we're seeing it now, but I don't know about you, Trish, but I see 4 arms.
Yeah, 4 for now.
4 for now.
There might be another one hidden back there somewhere.
One of the things you could see on that colony there, I should have said this when we were closer in, but you could see the exposed gold color of the Chrysogorgia colony below the sea star, which is an indication that that sea star has probably digested all the tissue moving up towards where the branches are. perhaps a little bit hard to tell because species in this family, the Chrysogorgiidae, tend to have very, very thin tissue on the skeleton anyway, so sometimes it's hard to see it, but you can clearly see the individual polyps arising from tissue on the skeleton, so you know there's tissues there. All those polyps are interconnected. But yeah, there was no tissue below that sea star.
So will a sea star like this just continue eating all the way up to the end of the stalk and then maybe fly its way down? It'll fall after it's done, or will how long does a sea star like this usually take to potentially eat a a coral colony that's this 1 meter potential in length?
Yeah, it's a really good question, and I'll bet that's Chris who just called in to help answer it.
Hey, this is Chris again.
Hey, Chris.
Hi,
Thanks
Chris.
for tuning in.
I'm sorry. I was calling in while you were asking the question. What was that?
The question was with the.
Question is,
Go ahead, Scott.
I apologize. Yeah, we'll keep feeding, and so it raises interesting questions, Chris, about how long do we have to sit and observe? Right, we would need to set cameras up, but you go ahead and tell the story of what we know about that.
Oh, you mean how long it's going to take? A long time, I think. Well, I mean, and this actually begins with data collected by Scott himself, because a while ago he observed. In fact, it turns out it was this species, Cerciaster. It turns out I think I jumped the gun when we saw it the first time. This is actually Cerciaster americanus, also a goniasterid, but very different from the Evoposoma that I was talking about earlier. But we identified Cerciaster feeding on a Paragorgia that had been knocked over, and Scott apparently came back almost a year later and found what apparently is the same two individual sea stars feeding on that fallen coral colony. So, I don't think it's reasonable. I don't think it's unreasonable to say that any one of these sea stars could be feeding on a coral like this for literally months. I mean, weeks, sure. Months, looking very good. But up to a year and longer, it wouldn't even surprise me if that was true. So they could literally just be living on the thing that they're eating. Oh, what's that? Oh, a little Chimopterus worm. Oh, how nifty.
That poly worm. Yeah, Chris, it was 18 months between those observations.
Okay, yeah. Did you ever go back to see that? I mean, after that?
Yeah, no, not on those two observations. So this was a situation where we had gone to Manning Seamount in the North Atlantic, and we had deployed some settling blocks, and we were lucky enough to have a follow-up cruise where we went and picked those settling blocks up a year and a half later, and that's why we were able to make these observations. It wasn't the intent that we were going to look back at these colonies.
Well, in that,
great.
I mean...
because now it's, 15 years, I think. I would love to see what's going on there.
Yeah. I get the impression that that's probably an important component of a lot of these ecosystems, is that their time scale is much longer than, a sort of visibly human one. And sea stars operate in an entirely different time frame altogether, because by our standards, they're quite slow. But by their standards, it's just right. So, and feeding doesn't take, feeding is very gradual because they extend their stomachs onto the polyps and digest them externally. But metabolism in these things is really slow. So, it would probably be a very long time, to take for one of these sea stars to actually completely demolish one of these corals. And frankly, that probably factors into the whole dynamic of, how long these skeletons take to, how long there's one of these little forests, one of these little oases, and, one sea star could remain on that one coral individual for quite a long time. and then move on to the other. So any one of these coral forests could last literally, decades, and it probably wouldn't be much of an effect on it in that time period, by our standards. So, but I mean, it would really be interesting to leave some kind of time-lapse camera and come back to one of these in, a year or 5 years even, if that was technologically feasible. Because so many of these animals have just a very different time scale than what we would be accustomed to, in terms of, just watching them move,
Go ahead, Lars.
A meter or a couple of meters along the deep sea floor could literally take,
cable rinser. Just wanted
months.
to do a comms check.
And so in that sense, it doesn't really make as much of a difference. And, people have talked to me about, well, why do you find all of these individual animals so widely separated across these distances? And, so in other words, why are there sea stars? Sometimes they're found only on their food, but sometimes they're out in the middle of nowhere on the abyssal plain. And people have just, said to me, well, why aren't they just moving across from one coral to the next? They don't have to be distributed by their larvae. They could simply be walking from one island to the next. And that's a very reasonable and plausible explanation. So when that sea star is done, it could literally just, clamper down from when it's finished and move across the bottom of the sea, take, 3 years to get to the next coral, wherever it might be, and get started again for its next 3-year feast. So it could be a 10-year journey just to have dinner. So, lots of interesting questions if we had a different time scale and the technology to watch them do it.
Hey, Chris.
Yeah, this connects back to, I think, a question that Tara Luke's class had earlier about the temperature and metabolism. It's cold down here, and we are living at a much higher temperature. We're warm-blooded. We burn through our food a lot quicker. We have to eat a lot more, whereas down here in the deep sea, they're adapted to conditions that are a much slower pace of life.
So
I apologize. We keep cutting off you guys on the ship.
Oh, no, you're all good, Scott. No worries. I was just about to ask Chris, speaking of these long time scales that these predatory sea stars might be making their movements across the sea floor or working on munching on one particular coral. Can you talk to us a little bit about what might be the lifespan of some of these organisms?
Oh, boy. A lot of the corals have literally been dated at thousands of years. And what that means for the sea stars that feed on them, I don't know. It doesn't seem unreasonable to expect that some of these sea stars are decades old. It wouldn't even surprise me if they were discovered to be, over 100 or older. We see different sizes, but growth in sea stars is not something you can measure easily because I've seen them shrink if they're not eating. So, and it's not clear to me what's going on, whether it's just resorbed, because they're not strict. The skeletons are endoskeletons. They're infused with tissue. So, it's not like strictly a matter of simply, it's not just like a matter of simply losing the calcium carbonate, like as an inorganic skeleton. They can presumably resorb some of that, and there can be contraction. But yeah, it wouldn't surprise me if some of the echinoderms are decades to maybe, and I have no basis for speculating beyond decades, but it is plausible to think of them as 100 years old. But that said, and there is evidence for sponges being thousands of years old, evidence for a lot of these corals being thousands of years old. So, again, the metabolism and the life cycle, can vary widely, especially since the larva of some sea stars, for example, is thought to last years in the water column. So it could have an entirely separate life before even settling down as quote unquote adults. And that could be the same for corals as well. I'm not exactly sure how long coral larvae can remain in the water column. Scott might be a better person to ask for that.
I don't know if that's a lead-in, but I was typing in it. I'm sorry. First observation of a purple what we call clavularia, this membranous or ribbon-like Octa coral is growing on the base of that white fan that we just passed. Yeah, Chris, good question about how long coral larvae can live. There's all kinds of different strategies for reproduction among corals, both shallow water and deep water, and it's one of the things that we really need a lot more study on in deep sea coral. Like a lot of things in the deep sea, it's challenging to be able to study. But yeah, for students who are out there and thinking about graduate school and what would be some good projects, thinking of some creative ways that you could study the larvae of these things would be really useful. So if we're lucky enough to, hang on a second, see what this is. Yeah, there's some more of that purple clavularia that's overgrowing a dead stalk down there. If we were lucky enough to collect a colony that was producing larvae, we would learn something about it by looking in the polyps and seeing, what's the stage of development in various polyps. But really, you'd like to be able to catch them in the water column, and that, as you might imagine, is pretty difficult to do. So this is a member of a group that's referred to as the Stoloniferous octocorals, and what that means is they grow like a ribbon or a mat on substrates, rock, or as you see here, an old dead skeleton. It could be on a sponge. And so a stolon, kind of like if you've seen strawberries grow in the garden, they put out these runners and individual plants can come up. It's the same sort of thing with this, except that it's like a mat that's glued to the rock. And so it doesn't create its own upright skeleton. Instead, it depends on other things that are already on the bottom. Lots of sponges showing up here as well, these white-blue structures that are close to the rock.
Beautiful view on Stream 2, Sirius.
So if anyone at home turns to Stream 2, you can see Sirius here with a really wonderful view, sort of getting an almost top-down view of Sirius in relation to some of these coral colonies, as well as these geologic structures that are almost seem like cut through the geologic feature.
Looks like there's also quite a few sponges here on the top left of the screen. Just really a lot of incredible life and biodiversity on this geologic feature here that we've been hanging out on all morning since the ROV is touched down. There's quite a lot to see here.
It's a really nice view of what we earlier tentatively identified as Hemicorallium beari. That name beari is B-A-Y-E-R-I, and it's named after Frederick Baer, or Ted Baer. He was a curator at the Smithsonian Institution in the Cnidaria group, and he's one of the most prolific octocoral taxonomists. He's just done incredible amount of work since the 1950s on describing lots of new species and understanding their biology. And sticking out behind it, on its left, there was kind of an angular coral. That was another kind of bamboo coral. Les Watling was just saying might be useful as a collection target, so we'll keep our eye out. We see that again. It's recognizable by pretty acute angle between.
All right, Control. This is Duck. How do you have me?
More organic curving branches that we've been seeing in the colonies.
Copy that. We're getting finishing touches
Thanks,
down out
Scott.
here.
Yeah,
you
if
guys
you
going to be
see
off
something
the
that is
off the winch for a little bit?
new in the area, new morphological
No, we're still climbing
type
up
or,
to the top of this
something
ridge
we're not
here,
sure,
so it's going to
let
be
us know
another
and we
few
can
minutes.
talk to pilots
Copy
about
that. We'll
potential sample.
All right, Lars, stand by one. We're getting in position.
Good copy. Standing by.
Looks like right in the center of screen right now we see a fish. Thank you. And pilots, could we get lasers, please? Thank you. I'm not sure what kind of fish it is.
I think this is a kind of cusk eel.
Okay.
Yeah, this individual looks pretty similar to several others that we've seen on this cruise as we've been making our way down the west coast of Florida through the Gulf of Mexico.
We saw some of the larger cusk eels during our wreck dive 2 of this cruise. These look much smaller than those that we saw before.
Can we get a look at the coral? It's just in the lower center. It's kind of pink-orange in color, and it's spiraling, so it's a type of Iridogorgia, just off the bottom. The coloration looks different.
Yes, so pilots, if we could, we have a request to see the coral that's now, I believe, right in center.
Going to be off-screen center for me. Yeah, there we go. Sorry, I'm a bit delayed, I guess.
All right, Lars, we're off winch. Green light. Please proceed.
Good copy. We'll set it up and then have you guys come in and out when we're done.
Iridogorgia is another possibility. I'm just noticing that it's a much deeper orange color or pink color than the ones that we've been seeing earlier. And we have Les Watling on the chat, and he's described, 3, maybe 4 of these species. So he's currently the leading expert on this group. We'll see what he has to say about it.
Yes, please. Yeah, it does seem to have that deeper orange color than we saw before with some of the Iridogorgia.
Yeah, and one of the things we look at is how big the coils are. And what you notice on this, I think this is a Rodan or Rhydigorgia, the coils, instead of forming a circle, it's more like a wave back and forth. And that was one of the characteristics of the Rodan or Rhydigorgia, but I don't remember that one also being this red or pink. This is quite interesting. I'm also noticing that the polyps look kind of tall and almost like they are lateral along the branch instead of poking directly off the branch. Uh-oh. So that's different. I hope Les didn't go to lunch. Beautiful shot there. You can see clearly the 8 pinnate tentacles I was describing earlier. Each of those tentacles has those 2 little side branches that come off each side. That's what we mean by pinnate. Each individual is called a pinnule. And you can see a bunch of polyps that look like just little mounds, and that's because they're able to contract the tentacles and withdraw them basically into the mouth. And so, I don't know if they're sleeping, resting, I don't know what's going on, they were disturbed. And I think a lot of them look kind of swollen, and I'm going to suggest that that means that they're in an advanced reproductive state. They're storing up gametes, perhaps eggs or sperm, and either getting ready to fertilize eggs, or perhaps some of them have been fertilized and the larva is developing. This is something that Chris was asking before, how long do the larvae live? We don't even know, are the larvae brooded for some period of time inside the polyp, or are they released, or the sperm and egg released into the water column and fertilization takes place there? There's so much that we don't know. So I see Les is here. Yeah, Les, do you have a guess? Would you agree that this is Rodan or Rhydigorgia? He is agreeing with me that the polyps are in a reproductive state. And you notice that most of those polyps seem swollen, so this whole colony looks to be having a synchronized reproductive episode that is synchronized among the polyps. And that's another interesting question.
I think we're ready out here. We'll go ahead and get the water on.
A few polyps are reproductive.
And then we'll, once that's running, we'll have you guys come in and out a little bit.
Thank you so much for that information, Scott.
Also, there's a couple of shrimp that are living in there. Swatlings identifying the shrimp, Bavipalymanella ceratophrons. Not unusual to see them associated with Chrysogorgiids. Anyway, this is in the same family as the Chrysogorgia and Metallogorgia we've seen earlier. All of them have this characteristic of having very delicate polyps on these thin skeletons. This one looks like it has, I think I'm looking at a very thickened part of the chelicid. That's the part of the leg that has the claw on it at the front there. It looks like it's quite inflated. Sometimes in the past, when we've seen these shrimp hanging out in the colonies, the coral colonies, what we've discovered is that they're females that are brooding eggs. They would be holding eggs on under their abdomen on the plesioids, which are the swimming legs. So presumably they're in there to protect their developing embryos. So yeah, let's suggest this Rodan or Rhydigorgia fragilis. It says that that inflated claw is like a pistol shrimp or a snapping shrimp, designed in the same way. This is not a snap shrimp, but it has that enlarged chelo. Beautiful stuff. It's always hard to take your eyes off of these Rhydigorgia and Rodan and Rhydigorgia colonies.
It always is hard, that's for sure.
So with shrimps like this, if we do see one sort of enlarged claw, is that is it usually just one? And is there a preference between the left and right? Or is this something that just depends?
Yeah, so I'm not an expert on that group, but I do know that at least in some of the species that you see that it's usually one claw or the other. And I'm trying to recall if there have been any studies, any genetic studies that try to determine if there is some relationship to which of the two claws is enlarged. I know that at least in some species it's related to sex, so the male will have the larger claw and they may be used in attracting females. I have no idea if that's the case for these deep sea shrimp that we're looking at here. And Les asks if there were two shrimp, and there were indeed. There was one near the top and one about halfway down. Chris is asking about a sea star in the lower corner. Let's see what corner he's referring to.
I'm not sure if I see that sea star. We might have already gone past it.
What about the little patch of sediment at about 3 o'clock? All the way over to the edge, there's a brownish blob over there.
All right. Yep, we do see that over to the now lower right corner of the screen. Would you like to have a closer look?
Yeah, let's see if that's kind of like maybe a bat star or something that he's referring to.
Pilot, this is Survey 2. Hi, could we get a closer look at that brownish black rock formation, or potentially rock formation, but it's a little bit square on the bottom right quadrant of the frame right now?
I think you may have identified it correctly as a rock.
A little bit too far away to tell from here, but as we get closer, it's looking a little bit more geologic than biologic.
Yeah, I probably missed what he was referring to. I was grasping at straws there.
Thanks, Pilot.
Looks like we have some more glass sponges coming into view, with some what looks like additional coral rubble down what would be probably bottom center. Looks like another Antipitharian, black coral. Wow, you see this coral colony with the Iridogorgia that's looks like it's been taken over by quite a few crinoids. And the bamboo coral here that looks like all of the polyps are gone.
That one's looking a little bit
Yeah,
barren.
there's something really interesting observation, and it is that you notice that the crinoids aren't on the live Iridogorgia or the live bamboo coral, and that's probably because the tissue is producing some chemical that acts as a deterrent for them to settle on. That's, kind of a best guess, certainly as a larva anyway. So there are definitely chemical defenses that are being used by at least some corals. So you can see some species, it's more routine that you see things settled on them, whereas other species, it's relatively rare. It's also instructive to see that once they die or if some predator has removed some of the tissue, then things will settle on the bare skeleton where there is no tissue, and that's a pretty good indication that there must be something going on at the tissue level that prevents settlement.
Sorry?
Can't tell if there's some snails or amphipods. There's a couple of things on that bare bamboo coral skeleton.
Pilot, this is Watch Lead. We were wondering if we could get a quick snap zoom on the stalk of that bamboo coral that's straight ahead, the skeleton, to see if there was anything on it. All right. Thank you. Oh, yeah, it looks like hermit crabs.
It's a nice spot, Scott.
Skeleton just wasn't smooth enough.
Oh, looks like there's a little brittle star. Is that an isopod? Looks like one, or a chitin.
I seem to have lost the internet temporarily in my office, so I didn't get to see it. Now I'm on. I can't help you.
So Les is letting us know that some of these Iridogorgia that we've been seeing along this dive seem to have a pretty good distribution of age amongst the different individual corals that we're seeing, but he hasn't been able to spot any truly small colonies yet.
The two squat lobsters that you see on that colony at the top we were just passing over, those are in the family Haverocideris. I kind of love that name because it basically points to their claws, and it means needle claws. They have these long, long arms with pretty narrow pincers at the tip. Is yet another kind of black coral. This is probably the genus Parantipathes. And one of the things you can see about this compared to some of the others we've been looking at is that the polyps are teeny. They're really small. And you can see what appear to be tentacles arising directly from the branches. And what it is in this case is that the polyps get stretched out along a branch. So recall earlier when I was describing it, I said that each polyp of a black coral would have 6 tentacles. But here, they look like they're arising in pairs, in twos. And that's because the 6 tentacles are stretched out. And so if you count, like starting at the tip of one of the branches, if you counted 3 pairs inwards, you would know, okay, that's the extent of one polyp. And the center pair of tentacles would have this little raised tube in between them, and that would be where the mouth is. Really bizarre little things.
As we're coming into this area, we see, yes, a very interesting geology video. It's seems to be not quite as smoothed around as before, with what also looks like a lot more coral rubble. We see more jagged edges and what sort of looks like almost little craters right in the center of the screen that just sort of sit.
The patchiness that we see in the deep sea is so interesting, how very quickly you can move to where we had so many colonies and such diversity to where we see so little. And so there are plenty of questions and hypotheses about, why you would see that pattern. Why is it that, just a few meters ago, it was a really good spot for the corals, and why isn't it apparently so good here? And some of the factors are the luck of recruitment. First, you have to get a larva that arrives in a place and settles and grows, and then it's got to be growing in the right place where, A, there's enough stability to the substrate that it can hold the colony while it grows over long periods of time, and, it doesn't get buried in sediments. There's quite a lot of sediment here, and so when you're a larva, you could easily get buried. But then there has to be enough current to provide food for you growing over the long term. No landslides or any other sort of things that would break up the colonies, the temperature of the water, how fast the water's flowing. There's so many different characteristics that going into whether or not these communities actually develop, and, is the subject of a lot of research. And this is why, these observations that we make with Okeanos Explorer are so valuable, because we get to see what is the scale of patchiness, and how does that relate to whether we're in a canyon or on a seamount or on a flatter slope and so on. And the more of these sorts of observations we can make, the more we can tie them together and make better hypotheses, predictions, and then do some experimental work to see if they're confirmed.
Yeah, thanks for that, Scott. It is definitely pretty incredible how we, in just a short distance, have gone from this really rich, dense area of biodiversity to what appears to be a lot more barren. So it's really interesting to know some of the drivers of settlement and recruitment of these corals.
Another great view of Sirius from the Sirius view on stream 2, sort of a ridge that we've come up.
Another thing to consider, you know, you were just talking about the drivers that lead to the community development, is the time scales, you know, kind of what Chris and I were talking about earlier. These communities, you know, have been here for thousands of years potentially, and so we're not just looking at what's happening now or last year or 5 years ago, but the result of processes that have been going on for centuries, basically.
So, copy. Thank you. So if those of you tuning in, if you turn to, I think it's stream 3, we have Blue View, which is on full view now, and you can sort of see what the pilot was discussing about looking to see the full view of this structure, this geologic structure that we're coming up on. Is that a fish also straight middle of the screen on? Is it a sponge? Oh, it looks. I did. It's a spongefish, yes. And also, if you turn to stream 2, you can see Sirius and how this geologic feature, we're coming up right on the crest of these two sloping sides. It's almost as if we're ascending to this peak that we're coming to. And it looks like we're coming into a nice little coral field of black corals.
And so this gets back to the patchiness question again. You know, we were talking about where do communities develop, and then what about the species that are making them up? So I agree here. There's quite a dense patch of these black coral whips, Aphanis stichopathes, and I would guess, if I had to, that this is something about a local recruitment phenomenon, meaning one or two corals got in here, found it's a good place to live, they were reproducing, and their offspring are settling very close to them. And that's why you see so many. You know, one possibility. That little fan in the background looks like maybe a little Promenoid right in the center. And I don't recall seeing much in the way of Promenoids today, which is unusual. That's one of the more common families in the deep sea.
Yeah, I think we had some of these Promenoids right when we touched down to bottom here at the beginning of the dive. It's nice to see them reappearing again.
And then several stalked crinoids. Those are always beautiful to look at, really interesting. Very closely related to the feather stars that we saw earlier sitting on corals, but they grow on their own stalk.
It looks like we might see some more of these either gastropods or hermit crabs on that skeletal structure of the coral there.
I didn't get to see them last time because my internet was not working at that time, but I heard you say hermit crab, and then you were wondering about something else you saw.
We were just discussing that we're seeing more hermit crabs that are on skeletal structures of these bamboo corals, and wondering, are they using that as a platform to get higher up to catch more of the particulate that is floating through, or is it more of just a hangout spot, or what's the advantage of moving further up of the skeletal remains from the coral?
Yeah, I mean, it's a good question. In general, most of the organisms that we see up on these corals are trying to get somewhat above the bottom. If you think about the physics of water flow, where water is in contact with the bottom, it doesn't move. Its velocity is zero. And as you move away from the bottom, it moves faster and faster as there's less and less friction with the bottom. And so if you are an organism that requires getting food out of the water, you want to be in that sweet spot where the water is moving fast enough that there's plenty of food that can drift by, but not so fast that there's too much turbulence or you're dislodged from your perch. And so corals and sponges are providing what we call this three-dimensional structure, this habitat structure created by an organism. It's basically a bioengineer that's building up off of the bottom that gives them a perch to capture things. So I don't know the specifics of the hermit crab, but I would guess that, it's up off the bottom so that it could, snag something out of the water. That's one possibility. Another is that it might be grazing on bacteria or something that's growing on that skeleton itself, or hydroids. There's lots of little things, worms and so on, that are living on the dead skeleton, so it might be using it as a food source. Maybe, if this were a salt marsh, there are various crabs and snails and so on that'll climb up on salt marsh at high tide to get away from predators. I suspect that's not the case here. I would suspect that the predators would be fish that would just come and pluck you off if you're off the bottom. So it's probably more related to food.
Thank you for that. Yeah, it does seem to be more advantageous, yes, to be further up in the water to grab the things coming by. So as we're finishing up this ship move here, we're seeing some of these sponges right at the top crest with additional black corals, and again more of the coral remains that are around the fossilized coral. And it does seem to have less sediment in this area, the sedimentation of settling. But it's the interesting sort of stepping of that geologic feature.
And if you're just joining us now from shore, it's been actually quite interesting over the past about hour or so. We've seen quite a big shift in density of these coral colonies and of these biological structures as we've moved about this geologic feature. So Scott France was on with us just a little while ago commenting on how different these ecosystems can be if you just move to the different side of a geologic feature. And there really is a lot to be learned and a lot of opportunity for research in this area so we can better understand what some of these environmental drivers might be that are affecting these changes in density that we're seeing on these features in such a small area.
Looks like we've got a big bunch of coral rubble with what looks like a pretty large piece remains.
Yeah, really interesting as we're coming around now. There seems to be a fracture in this geologic structure here with quite a bit of life on either side of it. So it looks like there's some sponges underneath that outcrop just above that fracture line area and a couple more coral colonies.
It's interesting that some of these coral colonies are growing outwards instead of outwards and upwards like we've seen some of the other ones do.
If you tune over to stream 3, got a photo that Scott France sent us of the class from the marine biology class from Mississippi College that was watching us earlier today. Just want to give a shout out to them. Thanks for tuning in, and thanks for being part of the exploration with us.
Yeah, and Dr. Toma there, who's the one at the far right in front, she was saying that she had prepped the class that, you might be spending the whole day just looking at marine snow, and so they're all really thrilled that we're in this pretty high diversity area in this canyon, so good timing for them.
Yeah, absolutely. And thank you, video, for the class.
So I apologize. I'm not sure if I'm listening to Anna or Trish.
So this is Anna.
And this is Trish.
Hi, Anna. Okay, thanks. Oh, you both sound so similar. Well, one of you was asking about or saying something about the way that colonies are growing out and then up from the wall. And one thing to think about there is, how do they start? And they start as a larva that's swimming around, has a very different mode of life from the adult stage. It's ciliated, and so it can move around pretty good. And it kind of probes the surface. We don't know all the details about what it's looking for or how it determines, this is a good place to glue myself down and then grow for the rest of my life. But if it's on a vertical surface like the edge of this wall or even under an overhang, it has to start growing, and then it has the ability to effectively sense gravity and is curling and then grows upwards so that it can get into a good current. And so one thing about growing on the flat surface or the underhang is that you're not going to be subjected to being buried by sediments in that location. So that would be one good strategy for a larva. But in terms of the bigger sort of habitat and scale of this canyon and these clefts and so on, I don't know what it is that larva is looking for, or how it knows the difference. It may just be luck, and maybe, you know, there are lots of larvae that settle, but there's only one or two or three that survive to get to the size that we're seeing.
So we started seeing a bit more of sponge diversity as we were coming in here with than we were seeing before, where it was mostly corals. And so we've seen a few up here, and then at about 12 o'clock, you can see there looks what looks to be another glass sponge, sort of hanging down.
Are we able to get a closer view? Oh, read my mind.
Nice little brittle star there for you. Big brittle star.
I believe that, yeah, this is one of the family Promenoidae, perhaps the genus Narella. Yeah, that brittle star is really tangled up and wrapped around there. Seems to be overgrowing a stony coral, mostly dead stony coral. There's a little bit of alive on the lower right that's white. That may be Lophidia.
There's just so much life on this wall.
I'm not a geologist, but it looks like, parts of the crust here have collapsed. You can see what appear to be, not a geologist, but it looks like, my bad, sorry, I had my computer on there. Yeah, you can see what look to be fracturing and some little sort of cave-like structures, and depending on the timing of when this part of the seafloor has collapsed, that could affect the corals growing there. And I just made a note in the chat room that I was seeing more dead skeletons on the floor here, and that could be the result of, some of the movement of the seafloor. I don't know if this is really, really long ago or not so long ago, but definitely some erosion and geological processes going on to affect the bottom.
Such a cool view from the Sirius camera.
And for those tuning in at home, if you have a look at the Sirius camera, which is on feed 2, I believe, it just really shows the scale of this feature that we're exploring today as D2 comes over the pinnacle of this geologic formation that has had a ton of life on it. It's made for a very exciting dive so far today.
a nice side view here of this, it did it again, sorry, of this Chrysogorgia colony in the center, the one that's kind of an orange color. And then behind it's a little bit harder to see in the darkness. To its left, there's I believe, an Iridogorgia colony growing there. And they're both in the same family, which is why I'm pointing them out. And one of the interesting things about the corals in that family is how geometric they are and regular in their production of their branches. And it's one of the ways that they're described. The angle of branches, the number of branches that come out as the main axis spirals, and various characteristics like that. And the name Chrysogorgiidae translates as golden coral, and you can see here why they're called the golden coral. You can see that gold color shining from beneath the tissue. It's kind of a iridescent.
Yeah, thanks for that, Scott. These colorations on some of these corals we've seen today are really so vibrant, quite beautiful. Yeah, you can see that Iridogorgia in that bottom left quadrant of your screen. Looks like we've got several brittle stars wrapped around the stalks of these corals here. Oh, wow. Thanks for this zoom here. This is just a really great shot of this brittle star wrapped around this coral. Yeah, we're seeing lots of brittle stars on these corals here. So get another zoom in.
Shoreside Kid, and anyone hear the telephone conference line at this time?
So we're just pulling back out here a little bit. We can zoom back out now and see some of this larger geologic formation again. Seeing some of those, it's really interesting, the structures that are sort of folded in on each other. Some of those black corals there in the middle of the screen. Looks like we're coming around on another really amazing coral feature. And as we've got these lasers out front, you can see just the scale of how large these corals actually are. And got a couple more squat lobsters hanging out on this coral here. Video, this is Watch Lead. Yes, we can hear you. Ah. We're getting a nice zoom in here on this squat lobster. And these look like big colonies of some sort of Corallidae.
Shore says, just so you know, we're having difficulty with the phone line. We're still trying to reconnect it.
So it looks like it's some sort of corallium or potentially a hemicorallium. And we've got Les Walting in the chat room telling us that based on how large and dense the polyps are, this is probably more likely to be a hemicorallium, Bayari, or I'm not exactly sure if I pronounced that right, but more likely to be a hemicorallium than a corallium based on the density of those polyps and how large they are. Just overall a really incredible feature of corals that we're on here. It's quite likely that these corals are very old based on how big they are here. So we've been chatting quite a bit today about the time scales associated with some of these organisms that we're seeing on this geologic feature. It's pretty incredible how large some of these coral colonies have grown to be. So now as we come around, you can see the backsides of these, what is identified as hemicorallium. Just really awesome structures of these coral colonies. And we've got some crinoids on the top of this hemicorallium here. It's really beautiful feather stars on the backside of the bottom coral colony and off the top of this one that's now in the center of the frame. And just an overhang in the geology of this feature in the background here. It looks like there might be another crinoid underneath it. Yeah, so Scott Francis in the chat room and has just commented that these coral colonies are aligned in the same direction. So they're facing into the dominant current here, which could, be, we were talking a little bit earlier about some of the environmental factors that contribute to the way these colonies form or how they settle and recruit onto the substrate here. And these corals have directed themselves into the dominant current. Oh wow, we've got some really spectacular crinoids here out over the outcrop of this geologic feature here. Yeah, these overhangs and the crust here are just spectacular. And it seems like there's lots of life that is taking advantage of these kind of juts coming out of the cliff overhangs here. It's really, really fantastic.
So for those of you listening at home, we seem to have some technical issues with our telecom line, so please stay tuned on that. We do still have our science chat open, so if there is more commentary that you'd like to pick out or have certain organisms that you'd like to see or features, please let us know via the chat line. We are currently troubleshooting that area now, so thank you so much for your patience. And please shout out if you have any other things that you'd like to see in the chat room. We also have CTube for annotations. So please make sure you log in also to CTube. Have any annotations there. Our current live stream through CTube is also having some issues now, so you will not be able to view the live stream via CTube, but it does still catalog what you are currently viewing on our streams. So you can use also that. It also catalogs all environmental data that is associated with all of whatever annotation that you end up clicking. All right, we seem to be looking at this gorgeous feature here, and sorry if you got cut off on the line before, but not sure how much was discussed before the line got cut off or we lost track of that, but we're seeing some beautiful coral in this area. We have lovely crinoids. I see also a purple coral, if we can get a snap zoom on that also quick. It seems to be also this purple coral at the base of that white coral there, but as we're kind of working around this little feature here, we still have that gorgeous coral community that we've been seeing throughout this dive.
Shoreside, if you guys, anyone on the science conference call, if everybody could drop the line and stand by, listen to this live audio, and once we reconnect, then we'll have you guys dial back in. For the moment, we need to have everybody drop the conference call connection.
Looks like there might be some sea star at the base of that Iridogorgia at the center bottom of the screen. A nice sea star at the base of that Iridogorgia. Thanks for the zoom. So Chris Maas identified this as a Cerciaster americanus, and he's done quite a wonderful job at eating a lot of the stalk of this Iridogorgia. And this coral has been identified as Iridogorgia splendens. With a really beautiful kind of shine on the coral skeleton here. Yeah, the sea star's put in quite a bit of work. Pilot, this is Watch Lead. Are you interested in taking a geologic sample in this area? All right, let me check in with our scientists and I'll get right back to you. Pilot, this is Watch Lead. We scientists have confirmed that a geosample here would be great. So for those of you who are following along on shore, our pilots are getting set up to take a geological sample on this location. So remotely operated vehicle Deep Discover has a couple of rock boxes, which I believe might be on camera 2 at the moment. So they are working on deciding which sample to take, and we'll be stowing it away in one of those rock boxes for its return to the surface. Copy that. So the pilots are getting up, or looking for a good candidate here for a geologic sample. So as they're kind of scanning around the area for what would be a good candidate, they're looking for something that's potentially already a little bit loose and able to be grabbed by D2's manipulator arm. So if you were listening just a few minutes ago, when we first decided to go for a geologic sample, some of the rocks might have been a little bit too compact without very many loose rocks available that would be good candidates for sampling. So we're just scanning around for a loose rock that would make a really good sample.
No problem. Thank you. Overall, good practice. All right. Pilot and co-pilot, did I hear that correctly? We're going to wrap up around here and try to... Oh, I'm hearing some feedback. Try to hit midwater? Okay, sounds great. Thank you. For those of you just being able to tune in, we did have some issues with our telecom line. If you would like to call in again, I will be posting a number right now that's temporary until we can potentially troubleshoot the other number in the science chat room. So I'll make sure to have that posted there, and you can then call in via that number following the passcode from that area. As our pilot and co-pilot stated earlier, we're going to be about 10 to 15 minutes still working around this more pinnacle-like feature with these corals, and then we'll start to ascend, stopping at various locations potentially for some midwater action. So thank you so much for all of your patience, and we'll go. We'll start to continue then this dive. Hi, did someone just join the call? Just checking in if someone joined in the call just now.
Thanks, Pilot. Yeah, maybe we can just get one last shot on that corallium, that white coral to the left, mid-left of the screen. Looks like we've got a crinoid, a brittle star, and a squat lobster all on top of each other, really close proximity on this coral here. The scientists in the chat room are remarking at how this hemicorallium is actually quite overgrown at the base from some other coral species. So if you're joining us from home, we are in our last few minutes of bottom time here before we start working our way back up to surface, doing some midwater exploration along the way. It's been a remarkable dive down here today. Seen quite a number of species of coral, crinoids, many of these squat lobsters that you're seeing in the middle of your screen right now, and quite a few brittle stars as well. This hemicorallium here is currently serving as home to several different species. And we are just doing a pilot change here before we start midwater exploration as we head to surface for the last ROV dive of the 2022 Shakedown expedition.
For those on the science call, if you guys have the new conference bridge phone number, we have the line reconnected to the new number. If anyone wants to dial in and use that, it seems to be functioning.
All right, for those who are still following along with us on the last ROV dive of the 2022 Shakedown, we have left bottom and are headed up in the water column from what has been truly a spectacular dive on a really lovely coral garden. We saw crinoids, we saw corallium, squat lobsters, brittle stars, just a really fantastic array of biodiversity on this fantastic feature that we found here off the West Florida shelf. So stay tuned with us as we continue to do some water column exploration while making our way to surface. And thank you so much to all of our scientists who have been incredibly helpful both in the chat room and on the teleconference line, helping us to identify some of these species and also understand a little bit more about the environment and ecosystems that we have been diving on today. Stay tuned.
Yep, that's correct. Just testing it out for you.
Yeah, so how's it sound, Scott?
It's a little bit, it's not as loud as the last line. And once in a while, I could hear this over the video too. I'm getting this weird sound.
Yep.
Like that.
Would you say the level is about the same, or too loud or too soft?
Right now, as you're talking, the level is just fine. About the same. But initially, I thought it was a little lower. Maybe I'd need to hear Trish or Anna, because that's who is usually doing the talking.
Okay. Well, we have this as a backup. We're not certain what's going on with the conference bridge at the moment, but we don't seem to be able to. It thinks we're still connected, so it won't let us reconnect.
Yeah, it's weird. I don't know if the information got passed on to you, but I could hear you and apparently it was Mark troubleshooting. It was like there was an open mic picking you up. So I knew the ship was connected somehow. So you were still there, but I wasn't hearing the main. I wasn't hearing science leads, right?
Thanks for helping us to test out, Scott, and for your patience while we troubleshoot this.
Yeah, we can. Are we on a free jar or an available jar? Okay, so the can number on that is incorrect. The readout, the light that's lit is correct. Do we want to switch to one of the available cans just right now? So that's one less thing. Of course. Copy. And we'll draw out, and then we'll do a normal midwater suction collection. Go ahead. Yep. Yep. Yeah. Yeah, I thought that worked well yesterday. Copy that, Watchley. Or, I'm sorry, Dive Soup. Go ahead, Workshop. Copy that. That makes sense, Workshop. Yeah, okay. So I think it's worth a shot.
It's not like having the arm on there that's going to be in the shot, but at the same time, it might be just enough. That's true. You can also like have the net out like up high or down low maybe and see if you can, what's underneath it. I guess.
A lot of options.
Well, we'll get there and see what's working and what's not.
Also, the other thing is when we had the net out, kind of dark the whole time. Like, what if you had the net out, had it light, and drive towards certain objects? Like, actively use it as a agitator rather than a passive, an active agitator versus a passive agitator.
Yeah. I think they're all good ideas. I think the one that jumps out at me that is, if we see something and we get it in frame, then, normally I'm drifting backwards or I'm pushing forward and then just reversing thrust, and that throws it around a little bit. And, that it was definitely, and then like the vehicle's moving in a direction then that, is going to make it easier at least to keep it in frame. That, so when I'm in auto depth, and that's how I kind of got in a little bit of trouble yesterday, taking out auto depth without a Z-bias really dialed. I don't know how much time we'll have. I mean, that might be something that we try, dialing in the Z-bias first. Because the autoverts are definitely, they're designed to get someplace and stay there, so they kind of, they're not made, you can't really use them to do, small movements like you want with mid-water work.
And so for those of you who are following along ashore, we are ascending right now, but we are planning to do some midwater exploration. Currently the target depth that we are heading to is about 460 meters, where it looks like the deep scattering layer is sitting at that target depth. So although we are ascending, we are not quite done yet with the last remotely operated vehicle dive of the 2022 Shakedown. So stay tuned as we ascend. We're seeing some ctenophores flying by, saw a shrimp fly by just a moment ago, so we are not quite done yet. Hoping to see some really great biology as we're moving up in the water column here. And also planning to probably look for some more bioluminescence, try to stir it up with that bioluminescence agitator that you've seen throughout the week as we've been testing that out with the ME20 low light cameras that are equipped onto D2. So stay tuned for some bioluminescence and some potentially awesome gelatinous creatures here in the water column.
someone increase the water on the rinser?
Nav, that target depth was 461? Thank you.
I think we start off with no education and work our way up from there. Kind of just come up with steps.
All right, well,
Like phase one thing at a time, you know.
that should make the whole operation simpler. Get my rotator at the proper angle. I think that's a great idea, video. Pilot's fine with that. Perfect. Yeah, let's keep the aux on.
Alrighty, so for those of you following along on shore, one of the things we've been working on quite a bit on this Shakedown expedition is trying to drum up some bioluminescence in the water column. So if you've been tuning in earlier in today's dive and for the past couple of dives, you've seen that bioluminescence agitator being used by ROV Deep Discover to try to agitate that water column and produce some bioluminescence in any potential organisms that might have that capability that are moving through the water column. So although we've just cut lights out on the ROVs right now. We're actually not deploying that bioluminescence agitator at the moment, are going to see if we're able to drum up that bioluminescence from any organisms in the water column just with the movement of the ROVs vertically as they ascend.
Copy. We can switch to ME20. Yep.
We are just getting switched over to the ME20 low light camera, which should be able to better capture any of that bioluminescence from the organisms that we are agitating as we move through the water column.
Saw a few. Like if the ME20 was mounted on Sirius, you mean? Oh, the tether would be agitating, yeah. Copy that. That's right. 461 is the depth. D2 crossing 550 meters. D2 passing 500 meters depth.
So as we're moving up in the water column, we are working our way towards the deep scattering layer, which is sitting at about 460 meters right now. So this deep scattering layer is a horizontal zone that typically has quite a few living organisms in it. We expect to see quite a bit of biology in this area.
Yes, please.
And we'll perform a transect here.
All right, co-pilot, you want to... Oh, you got a little bit more to go. Okay. Copy. D2's holding at 465. I'm going to put in a mildly believable Z-bias. Engage it. All right, first things first, we take that, well, I guess I'll just, as we deploy, I'll swing to port, and that'll put us back at a half turn. And then when we stow, I'll continue to port, and that'll take it all out. Okay. Okay. All right, auto heading engaged. And we got a good delta. I am backing. Looking good. Thank you for the Titan view.
So D2 is currently at 466 meters, which is the bottom edge of our deep scattering layer. And so as of right now, D2 is just going to be...
What's that now? Okay. I mean, I'm almost 100% aft right now too. Copy. Then I'll get a little bit farther out, and then I'll do my turn to port, and we'll be back at a half turn. Okay, gonna make the turn. Oh, all right, I'm gonna go the other way. Tether looks good. Okay, pushing forward. Copy that. I'll get a little bit farther out, and then I'll go off stick, and we'll see what it's looking like. Okay, I'm off stick. Compared with yesterday, I feel like I'm drifting aft slower, but strangely the particles seem like they're coming at me faster. That looks pretty nice though. I mean, what do you think, Nav? Should we try and slow it down, co-pilot? All right. Yeah, I mean, let me get set up one more time, and we'll check the directions. I on that drift backwards, I felt like they were. There's jelly.
Oh, it's a nice jelly there in the middle of the frame.
Yeah.
Looks a bit similar to the jelly that we collected as a sample the other day.
Yes, which was
Solmissus.
Solmissus.
So we're just switching over to that low light camera again, the ME20, to see if this jelly that we just saw in the middle of the frame here might have some bioluminescent qualities.
I'm drifting back pretty fast. I'm going to push forward a little bit. We're probably going to, yeah. All right, I think we're going to have to do that move, Nav. Okay, I'm going to push forward. As far as lighting goes, I'd say whatever you think works best for video. Oh, that subject's long gone, I think. Yeah. Oh, yeah. Easy. Yep. Yeah, we can, I think we can dim quite a bit from there. Yeah, that's good. Maybe even a little, maybe cheat a little dimmer.
So what our pilots are trying to do now is locate an organism
that makes it easier for video.
Cut the lights to see if it is already luminescing, and so we can instead not use the agitator and see if we can just capture something that is currently within our field of view. And there's a jelly right up ahead. So let's see.
Oh, this looks like a ctenophore here.
Okay,
a comb jelly.
A little bit of time on this.
So it was originally in the bottom left corner of the screen.
Yeah, I think we lost that one. I've got to push back out. And then when I get farther out, co-pilot, would you rotate back to 4-5? I sure do. I think that's heave. I think a heave of 4 isn't, well, I guess not, yeah. Yeah, I suppose so. I feel like my depth has been pretty solid. What? Okay. All right. So we got to keep a close eye on the delta. And Nav, if you could help with that too. Yeah, that's what I'm thinking too.
So what you're seeing right now is kind of the essence of why we do a shakedown cruise. It's our pilots are trying new things, trying to see what works and what the best methods might be for trying to observe, in this case, bioluminescence. So we do these shakedowns at the beginning of the field season to take an opportunity to try new things. Looks like a little fish moving around. To see if they might be feasible to continue with for the rest of the field season.
Oh, yeah.
So right now they're trying to find an organism that might bioluminesce on its own without pulling out the bioluminescence agitator. Small fish over there to the left.
Yeah, video, as far as the lighting goes, whatever works best for you. Okay, I'm pushing forward. Out. Here, it's some holidays on the USBL.
There's definitely quite a bit of life in the scattering layer here.
Once we get up to speed on that move, okay. All right, this is me holding position, and that's about 40% forward. Yeah, that's what I was thinking. It might kind of slow things down a little bit more. Yeah. Well, I could vert up to it, or you could rotate the pan and tilt up. Actually, I can vert up. Yeah, we'll grab the next one.
Do you mind if I try and pan up and see how much?
Sure. Yeah, go for it. Okay. Okay. Yep. Okay, we can. Okay, I don't have a ton of time here. Okay. All right, I think we might have lost that one. I'm going to push forward.
There was a little bit
Yeah,
something at the bottom there.
we'll get stretched out, and that way we can have a little bit of time to try and dial things in once we see another subject. All right, a little bit more. Well, that's some pretty dramatic heave. All right, let's get lights on. Well, stand by. Looks like Sirius heading was swinging pretty dramatic there. All right, that sounds good. All right, I'll drift back. I'll kind of waggle that beam, see if we see something. Copy that, Nav. What about Sirius? That makes sense. Well, that's right where we want to be, so. Ooh. Oh, it's just marine snow. Oh, perfect. Thank you, Nav. So I think you're right, co-pilot. I was too far out in front. Oh yeah, this is great. This is perfect, Nav. Thank you.
So if you're listening at home, what you're listening to right now is our pilots working together with our video engineer to try to figure out the best combination of lights or best combination of lack of lights to be able to see bioluminescence on either the M820 low light camera or on the standard camera on Deep Discover. So by trying to work out what the best lighting scenario is to have enough light to be able to see our subjects while we are working on this, trying to work this out, but also have the lights be dim enough to be able to actually see that bioluminescence in the water column. So if you're listening, you're really listening to just trying to figure out the best case scenario for combinations of lights and cameras.
Well, if everything was the same as yesterday, I think we were definitely seeing a lot more with the agitator. I've been pushing forward and back, but no, I've been trying to do the opposite really. But that's a good idea, Nav. I can kind of shoot it around sometimes.
There's a jelly right to the bottom left corner that is slowly going past us, maybe. Yeah, it was right in front.
Okay, I'm going to back slowly.
Let's see if we can find it again.
Yeah, I think we were seeing a lot more luminescence with it than we have so far today, or without it. Maybe we can keep it in like half frame or something like that. Yeah, that's, yeah. Yeah.
Well, upper right.
Oh, right at the top at around 1 o'clock, there looked to be a strand of something. Ooh.
Yeah, so here's just threw a whole bunch of water out in front of me. I got to push out a little bit. I'm starting to fall back. Okay, I'll try a little bit more of that. And we can carefully change our depth. Watch lead, this is directed at you, this pilot. Right now, it's not, if something's like right above or below us, it takes a while to get D2 up or down there. And if we're chasing something like that, we can kind of get into a dangerous situation fairly quickly. So we can try it in a limited capacity, but I just wanted to, share that.
Yeah. Copy that, pilot. Thanks for sharing. Great.
Oh, did you see those? Okay. 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 Yeah. What are you looking at? What?
So if you're tuning in at home and you're wondering why we chose this particular target depth to explore,
a bigger object,
so this is what is called the deep scattering layer. It's currently sitting at about 465 meters in the water column, and this layer is just a very, very dense layer full of all sorts of mesopelagic organisms. So this could be anything from small fish to zooplankton to some of those jellies and ctenophores like what you saw a little bit earlier.
Workshop was saying that's
so something that's really interesting and unique about the deep scattering layer is this entire layer actually performs what's called a diel vertical migration each day, sometimes called the greatest migration on Earth. So this is actually a movement of the entire layer to shallower depths in the water column, moving towards the surface at about dusk for some of these organisms to feed on the phytoplankton at the surface, and then a movement back deeper into the water column during the day in order to stay protected from potential predators. So this migration happens every day, and it is incredible that this is happening in the water column, which is one of the most unexplored. We know very, very little about water column ecology, but it is pretty amazing that this occurs every day.
I was kind of worried with tilting, when you're changing the lighting. Okay, there's a fish. Ooh, that was something. Saw a trail. Okay, I'm going to push forward.
I think you guys want to brighten it up so you can see if there's something out there. Right?
Yeah, go ahead, Nav.
Yeah, let's get set up back into recovery mode. Next, it's time to head on up.
Copy.
We're going to hold our position once we get into it, and then we're going to kind of off to the southwest here.
Okay.
Around into the wind. What that means for you, pilot, is that you're going to hit that port anyway. It's not going to come all the way to the road. You're not going to come all the way to Oxford ship. You're going to face roughly southwest.
Copy that. And that's how we should set up for right now.
Yep.
Okay.
So I would say 225 or so.
Okay.
And you got to come to port anyway, I believe, once you turn out.
Yep.
And then Cirrus will turn to face that direction while the ship, and then the ship will slowly make its way around.
Okay, so while we're setting up, should we keep... Yeah, this is pretty safe. I was just wondering if we should do any ship movement changes while we're...
No ship changes right now.
Okay.
Chris is setting up right now.
Copy.
So we're going to just keep right now in our state.
All right. Standing by. I will head to 225, so you will be at 45. And like, Nav, should we just hold?
Yep, we're just going to hold. Once you get into tow configuration at that.
Okay, so we should get into tow.
Yes, but we're not coming out.
All right. Yeah. All right. Yeah. Back up a little bit more. Well, at least we're not seeing tons of jellies all around us right now after we turn the lights on. Oh, well. Okay, I'm coming around to 225. Okay, so you're coming around?
If you turn to string 2, you can see the view of Sirius, from Sirius, and there is what looks to... Oh, there it goes.
Now I'm pushing.
What looked to be a jelly of some sort, or ctenophore.
And something's holding you a little bit off heading. This is kind of a little bit of a weird...
Our pilots are just getting dialed in right now to begin their ascent to the surface and to wrap up the last dive of the 2022 ROV and mapping Shakedown.
All right. So there's a current
If
pushing me
you're
back.
still following along with us here,
while I need
we're
to
still
push
seeing
forward,
some
so I want to
really nice ctenophores and jellies. The
keep
water
pushing
column here at about
starboard as I go
465
forward
meters of depth, still in that
with
deep
this
scattering layer.
towing into these directions. Yeah, I think maybe we should have gotten in tow and then, yeah. All right, but we're almost there. All right, I'm in view. Co-pilot, I'm coming up. Okay, I'm disengaging auto heading. Okay, keeping it in. Yep. Copy. No, I think they wanted us to hold this depth. Okay, I'm engaging auto depth. I have auto heading. Yeah, I've got, all right, copy that. Got a little forward way on. Yeah, I have to lateral to stay in your tail cam. Yep.